see this one.
Finally, it's starting.
What you are about to see is not architecture as you know it. It is the design of life itself. When we think of architects, we imagine people designing cities, buildings, and bridges. But what if someone could design the tiny molecular structures that make life possible? This is where David Baker comes in.
What if we could design proteins from scratch, just like architects design buildings?
David Baker is an American biochemist and computational biologist who has pioneered methods to design proteins and predict their three-dimensional structures. Traditionally, scientists studied proteins that already existed in nature, but Baker took a revolutionary step further. He used computers to design entirely new proteins from scratch. By predicting how amino acids fold and interact, his work allows scientists to create proteins with specific shapes and functions before they are ever made in the lab. This breakthrough transformed chemistry, biology, and medicine, and in recognition of this achievement, David Baker was awarded the 2024 Nobel Prize in Chemistry. But to understand why this discovery changed chemistry forever, we first need to look at the problem scientists faced before protein design was possible.
What kind of problem could it be?
For many years, protein research meant studying only what nature already made. Scientists observed proteins found in living organisms, but they could not easily create new ones. This method had clear limits. First, it was very slow. Protein design depended on trial and error in the laboratory. Scientists changed small parts of a protein, tested it, and hoped it worked. This process could take a long time. Second, protein folding was hard to predict. A protein must fold into the correct three-dimensional shape to work properly. Even a small change could cause the protein to fold incorrectly and stop working. Because of this, designing new proteins was once thought to be nearly impossible. Proteins were seen as too complex and unpredictable to control. That breakthrough came with David Baker's computer program, Rosetta, which made it possible to predict and design protein structures before they were ever made in the lab.
That's like designing life on a computer.
Rosetta changed how scientists approached proteins. Instead of relying only on slow laboratory experiments, scientists could now use computers to simulate how proteins fold and behave at the molecular level. By applying the basic physical and chemical rules that govern how amino acids interact, Rosetta can test millions of possible protein shapes inside a computer. It predicts which structures are stable, which will fold correctly, and which are most likely to work. For the first time, researchers moved from guessing to designing. Instead of asking if a protein might work, they could ask what kind of protein they needed and let the computer design it. Rosetta transformed protein science into a design-based discipline where ideas could be tested digitally, quickly, and efficiently. This shift opened the door to entirely new molecules, proteins that had never existed in nature before.
Whoa, that's so cool.
So what makes this discovery truly groundbreaking? To answer that, let's explore the scientific significance of David Baker's work in computational protein design. Every architect must understand their materials. For the protein architect, those materials come from chemistry. Computational protein design merges chemistry and biology at the most fundamental level. Proteins, like buildings, are shaped by chemical forces, hydrogen bonds, electrostatic interactions, and hydrophobic effects that determine their structure and stability. Using Rosetta, Baker can design amino acid sequences that fold into precise three-dimensional structures. This control over structure dictates function, allowing creation of enzymes with specific activities and signaling proteins with precise cellular targets. His work transformed structural biology, advanced enzyme engineering for medicine and industry, and revealed fundamental folding principles, reducing trial and error experimentation. In short, computational protein design shows how chemistry, guided by computation, can predictably shape biology, reshaping modern protein science. It represents a new paradigm in molecular science, where chemical principles are used to engineer proteins by design rather than discovering them by chance.
So, can protein design really be used in real life?
At this moment in the story, the architect steps beyond theory. David Baker's blueprints are no longer confined to the screen. They are ready to change the real world. In medicine, the protein architect designs structures that protect lives. Baker creates protein scaffolds that act like the framework of a building, holding viral components in a stable and precise form. This allows the immune system to recognize threats more clearly, making vaccines stronger, safer, and faster to develop. But great architects also design with precision. In cancer therapy, Baker's proteins are engineered to fit only specific cancer targets. Like a custom-built lock and key, these designs block harmful signals while leaving healthy cells untouched. The story then moves beyond medicine, facing a world overwhelmed by plastic waste. The protein architect redesigns nature itself. Engineered enzymes break down plastics into reusable materials, transforming pollution into opportunity. Through these real-life applications, David Baker shows that proteins are more than molecules. They are structures and the foundation of a better future.
That's impressive.
I didn't know proteins could do that.
David Baker's influence can be clearly seen through his scientific publications. His research has been published in leading journals such as Science, Nature, and the Journal of the American Chemical Society. A notable example is his work on de novo protein design, where new protein structures were successfully designed with specific functions. These studies prove that computational protein design is not just a theoretical idea, but a practical and reliable scientific approach used in real laboratory experiments worldwide. Looking to the future, the impact of Baker's work is expected to grow further. The combination of artificial intelligence with protein design will allow proteins to be created faster and with greater accuracy. AI models can analyze large protein datasets and work together with tools like Rosetta to design improved enzymes and therapeutic proteins. In addition, advances in this field may lead to personalized enzymes tailored to individual patients, improving treatment effectiveness and reducing side effects. In conclusion, David Baker transformed how scientists approach proteins. Instead of studying only what nature provides, researchers can now design new proteins with specific structures and functions. By combining chemistry, biology, and computation, his work opened new possibilities in medicine, industry, and environmental technology. This lasting impact is why his contribution was recognized with the Nobel Prize in Chemistry.
That was awesome.
I will definitely find out more about this later.